Good evening, everyone. My name is Meredith Toohey, and I'm the Conferences and Events Manager for the American Nutrition Association, the ANA. The ANA would like to thank Bragg Health Foundation for their support of our 2018 Global Webinar Series. Learn more about their mission to inspire and educate people to adopt a healthy lifestyle through optimal nutrition, exercise, positive attitudes, and spiritual wellness at bragghealth.org. The American Nutrition Association is very pleased to be hosting Dr. Daniel VanDorn tonight to explore resilience and the role of nutrition. Let me take a moment to tell our audience about the ANA. We are a nonprofit organization which for over 40 years has been on a mission to promote optimal health through nutrition and wellness education. We educate the public and professionals in many ways, including hosting world-class nutrition programs such as this one tonight. We encourage all of you listening this evening to visit our website at theana.org for an abundance of nutrition information, resources, and upcoming webinars. Join the ANA as a member and help us continue to bring these nutrition luminaries to you. And now, we are honored to introduce Dr. Daniel VanDorn. Daniel VanDorn, PharmD, is a medical research scientist at Genomind, where he provides counsel and education to clinicians and patients on personalized wellness and psychopharmacology. Additionally, he is heavily involved in new and ongoing product development. Informed by his experiences in the armed forces, where he earned multiple awards and commendations, and disaster relief efforts in developing countries, his recent work has largely focused on mechanisms of stress resilience and the biomolecular underpinnings of cognition, mood regulation, and response to trauma. Welcome, Dr. VanDorn.
Thank you, Meredith. Thank you for having me. It's an honor to be here and speak with your audience. And hopefully I can add something to the lexicon and help support the connection between nutrition and health. It's something that is very near and dear to my heart. Now, I've done a few of these types of talks, and particularly for nutrition, and it seems somewhat counterintuitive. I've actually had this question before, that I'm a pharmacist, and I've even heard the term pill pusher before. And so I would say, first, no, if you have ever worked with pharmacists, particularly doctors of pharmacy, our focus is really reducing the numbers of medications that patients are on, or at least using them accurately, correctly. And in my clinical experience, the focus has always been on the modifiable lifestyle factors to help support that, to get patients off metformin, for example. And aside from that, I've actually, unlike most pharmacists, I'm actually more of a geneticist. My focus has always been on research. I'm more of a geneticist than a pharmacist. But my focus has always been on the biomolecular mechanisms, and particularly when it comes to stress response, which has been informed by my experience in the armed forces. I served in the U.S. Navy from 1998 to 2005, and then in multiple clinical settings where I saw the effects of trauma. And it has always been fascinating to me in how some people, or you take two people who are exposed to horrible events, trauma, just horror, and some patients or some people will be able to walk away from that and not actually have any adverse sequelae, while others will have PTSD and trauma associated, basically a mood dysregulation associated with trauma. And so that has been my focus. My research focus has been looking at what confers that resilience or that sensitivity to stress. And so my focus has always been on the genetic markers, the single nucleotide polymorphisms. And this is what I'll discuss with you today. But my focus will be tying that in with the mechanisms by which it confers its phenotypic impact. We'll talk about the brain circuitry involved with stress response, the HPA axis, and how it involves the brain-gut axis, as well as cardiovascular and inflammatory pathways to drive the risk. Because by focusing on the mechanism, it provides an opportunity for healthcare providers, in particular nutritionists, because you're at the forefront of both preventative and healthcare to prevent or mitigate these risks. And so we'll look at how to target these mechanisms with lifestyle modifications and dietary and supplementation to mitigate the risk associated with stress. Now, I use the term stress resilience. I think it is important to define that term because it is thrown around a lot these days. And so in the context of this presentation, resilience is referring to just what I was describing with those two people exposed to horrible trauma. And the story that I defined would imply that the subject who was able to walk away without any adverse sequelae had some type of inherited or genetic resilience to stress. And that's often what, when we're talking about stress resilience, that's the common analogy used. But there is another view, which is that resilience can be learned through training. In fact, that's the whole point of military training and boot camp, is to expose you through the rigors and through repetition, not to basically have that type of response to basically reduce your sensitivity. And like the nature versus nurture argument, these are not mutually exclusive. It's really a combination of the two. In fact, I'm going to go a little bit off the beaten path here. Whenever I do a talk on genetics, I actually like to start with a story. And the story goes like this. Long ago and far away, that's just how I like to start stories, there was a young man, a boy born without sight. He was blind. But he had all his other faculties. He was very intelligent, and so he could do many other things for his village, and he had a place of honor. And one of his favorite jobs, one of his favorite duties, was to count his family's cattle as they were leaving the pen to go graze and when they came back at night. And he always did it the same way. He was blind, so he couldn't see them. He would feel them. And as they were leaving the pen, he would feel the nose and the flank and then the tail. The nose, the flank, the tail. And he'd count them that way. And he'd do that morning and night, day after day, year after year, always with a look of rapt attention on his face, like he was trying to solve a riddle, because he was. But only he knew what that riddle was. And no one really could figure out what he was doing or why he was so interested in this until, years and years went by, and on his deathbed, surrounded by his loved ones, he was still thinking about feeling the nose and the tail. And then he said, aha, I understand now. I get it. The nose causes the tail. Now, I tell that story a lot, and I never really get a laugh, so I apparently have to work on my delivery. But it should sound a little funny to you, or at least strange, the concept that a nose causes a tail. We know that a nose on a cow does not cause a tail. They are associated. Most cows that have a tail also have a nose. But then look at it from his perspective. The way he always saw the cattle, because he lacked sight, is by feel. And for him, the nose always preceded the tail. And so he fell into a very common logic fallacy called pro hoc ergo propter hoc, before, therefore, because of. And I guarantee you that everyone listening to this program, the audience members and most scientists absolutely, have fallen into this trap before. And there is no field of science that is more susceptible to this error of logic than genetics. I will be discussing today several genes or genetic variants that are associated with a disease or a mood disorder or a stress response. But it's not that you should not interpret that to mean that these genes cause disease. They don't cause disease. There is no schizophrenia gene. There is no depression gene. Genes don't encode for disease. They don't express disease. If they did, we wouldn't be here. We wouldn't exist as a species. Genes encode proteins and enzymes and receptors. And these enzymes and receptors, they allow us to interact. They allow us to do stuff with our environment. They allow us to consume and absorb nutrients from our environment and metabolize them and break them down into other molecules that we can use for our body and excrete them. They allow us to regulate our diurnal cycle, to sleep when we need to sleep and recharge. They allow us to interact with our neighbors, to socialize, to fall in love, to taste, to feel, to lick, basically to live, in essence. But as a geneticist, my interest is those single nucleotide polymorphisms, those small changes in the genetic code that can upregulate or downregulate how much a gene is expressed, how much of that protein or receptor is made, or changes the size or shape of that protein that impacts how we interact with our environment. Now, the focus of today's discussion isn't so much those single genetic variants, but rather how do they impact that interaction between our environment and our cells. In essence, we'll be discussing epigenetics. And my focus, so what I identify, are the changes in the genetic code. But this is not something we can change, at least not yet. This is what we inherit. And this is not deterministic. Our genes do not define us. There are things that we can do to control how these genes are expressed. And that's where we get into epigenetics. Epigenetics is epi is on top of genetics. And this is how the expression of these genes is controlled. And when we talk about epigenetic modulators, in essence what we're talking about is what we do in life. Drinking water, interacting with our neighbors, eating the food that we eat day to day. Even now, if you're listening to this program, my voice is being propagated through like electronic media into sound, which is entering your eardrum, turned back into an electronic signal, and making changes to how your DNA is expressed. Just by listening to this program, you are changing your DNA and how it works. So again, the focus is not on how these genes cause psychiatric disorders. There is no schizophrenia gene, but rather what is the interaction between the gene and the environment that causes a phenotype. Another concept I'd like to introduce, which will come into play later, is a systems biology approach or a multimodal approach. Geneticists, I can attest to this, tend to get very granular, and they like these linear stories when it comes to genes. To digress one more time, I'm going to come back to this, is one of my favorite authors actually said that we missed the mark when we named our own species. We called ourselves Homo sapiens, which was somewhat egotistical, it seems to me. So Homo among the hominids really puts us closer to the apes, which are large and strong, but phylogenetically we're actually closer to chimpanzees. And then the sapiens, so Homo sapiens, sapiens means wise or wisdom. So we called ourselves the wise ape. And you really just have to turn on any 24-hour news cycle to see that we haven't done much lately to prove that we are very wise. He contends that we would have been better off and more apt if we called ourselves pan-Narans. So pans would be a chimpanzee, and Narans would be story or storytelling, the narrative, the storytelling chimp, because it's how we see the world. It's how, what am I doing today but telling you a story, perhaps several stories, to try to prove a point, to try to convey information. It's how we learn. We particularly like linear stories. We're almost addicted to linear stories, the pro hoc ergo propter hoc, the nose causing the tail story, because it's simple. We can fit it into our brain. But when we're looking at nutrition, when we're looking at health biology, when we're looking at systems biology, we have to use a bit of metacognition. We have to be very careful not to fall into those traps. And why I bring this up is we like to look at these genes as doing one thing. But the truth is nature is very parsimonious. It never makes, parsimonious means frugal, very cheap. It never makes a protein or enzyme receptor that does just one thing in the body. That would be a waste of energy. So it makes these receptors that impacts many aspects of health. Genes that regulate, for example, gastrointestinal absorption or balance of the microbiome, this dysregulation may present itself as anxiety, depression, or anemia, or cardiometabolic issues such as obesity, oftentimes tied in with inflammation. So I think as we go forward, we will discuss these genes and how they impact stress response, but the how they mitigate that risk, or actually how they confer that risk, not mitigate, will follow different pathways, different mechanisms. And that's really the focus here, is the mechanisms, not the genes, but what is the mechanism by which that variant confers a risk? Because that provides an opportunity to target that pathway to mitigate those risks. So one well-known enzyme or gene, I think, to the audience might be MTHFR, methylene tetrahydrofolate reductase. So this is an enzyme responsible for activating folate. It's involved in epigenetics, and so if this pathway is abrogated, you can have elevated homocysteine, which is associated with cardiovascular disease. But it is also involved in providing the raw materials to make catecholamines like dopamine, serotonin, and norepinephrine. It actually is associated with mood dysregulation and cognition. And we'll come back to that, but I just wanted to touch on this briefly to give you an idea, the general philosophy of this presentation. I wouldn't have time to talk about all the genes involved with stress, but I'm going to talk about a few that may be new to you and maybe not. Genes that impact the HPA axis, which is our fight or flight response, our sympathetic response to stressors in our life. Genes that impact serotonin signaling in the brain, which, not surprisingly, also integrates with the HPA axis, as well as cognition. And so we'll talk about genes that impact cognition, a sense of spirituality, and even excitatory signaling itself. And I will later tie this into how this may impact the risk of inflammatory pathways, which is associated with many of these neurological sequelae. Now, when we consider the stress or stress response, this is generally what we're thinking of. This is the picture we have in our mind. The resilient subject who was exposed to trauma and walked away and didn't have PTSD, this would be how we might view them. They have this nice sturdy rope that can take a lot of pressure and does not snap. But someone who is sensitive to stress, who is non-resilient, may have some nicks in the rope, whether this is genetic predisposition to hypersensitivity to stress, or environmental toxins, lead toxicity, or a mold, or prior stressors. Poor nutrition definitely has been associated with a poor stress response. So if you start off with this predisposition to stress reactivity or non-resilience, then you're less able to deal with the stressors of life. And we can see this in real life. So this is a graph that shows that with increasing rates of childhood trauma, both sexual and physical abuse, it confers a risk of increased depression, adulthood depression. BDI means Beck's Depression Inventory. It's a rating of depressive symptoms. And this shouldn't be surprising to anyone. But if you then overlay this with genetic information, this is the MTHFR gene that I discussed before, methylene tetrahydrofolate reductase, which is the enzyme that creates methylfolate from our dietary folate, because dietary folate is useless in the body until it's activated or methylated. This is the rate-limiting enzymatic step. But if you have a genetic variant that confers decreased enzymatic activity, so decreased ability to use dietary folate, decreased ability to make methylfolate that gets into the blood, into the brain and makes dopamine, serotonin, with increased rates of childhood trauma, it can increase your risk of depression up to twofold. But in the absence of trauma, there is no increased risk. So again, this variant is not causing depression, but it does make one more susceptible to life stressors to increase depressive symptoms. So that's a fairly straightforward story, and it would be great if it was that simple. But unfortunately, there's other phenotypic associations that don't fit this model so easily. So we have to refine our story. And so a perfect example of this would be the serotonin transporter. The gene name is SLC6A4, which is basically it's a serotonin pump. This is expressed in neurons, which neurons, the presynaptic neurons, when they release serotonin to activate the next neuron, you then want to transport that neuron and recycle that serotonin back into the presynaptic neuron so it can be broken down or reused. If patients, however, have this S allele, which stands for short, is basically a deletion which confers decreased expression and thus increased serotonergic tone, and they have increased rates of childhood trauma or stressful life events. Similarly, this graph is actually showing you a probability of suicidal attempt, but we'll see this consistently across the board, both depression, PTSD, anxiety, that those with a short genotype, so they're homozygotes for reduced expression of the serotonin transporter, they have increased rates of depression, anxiety, increased suicidal ideation or attempts. And if you just look at the right half of this graph, that does fit our stress diathesis model, that the nicks that occur in one's rope that makes one unable to carry the weight. But then look at what happens at the other side of this graph. With lower incidence of life stress, so an enriching environment, the inverse occurs. Those with a short genotype actually have fewer depressive symptoms, fewer rates of ADHD, fewer suicidal attempts compared to those with the normal, I should say, quote unquote, normal genotype. So this is interesting. This does not fit our model, and this has been replicated across multiple studies. So we have to refine our model, and this is where we get into the differential susceptibility model. And I like to sort of define this, and I tell this story during clinical consults. It's the difference between an orchid and a dandelion. So I'm a bit of a green thumb, and I do take care of my own lawn, and I love orchids. I think they're beautiful flowers. But if I ran over an orchid with a lawnmower, I'm not going to have much luck in bringing that back to life. I'm going to have to go out and get a new orchid. However, dandelions, which I'm not so fond of, and I realize my audience may be raising their eyebrow at this, I realize that dandelions have nutritional and medicinal value, if properly blanched and all that, but as far as aesthetically, I think of them as the rats or the pigeons of the horticultural world. But when I try to destroy the dandelions growing on my lawn, the very next day they're back in force. I just can't get rid of them. They're very resilient. And so that would be like the right-hand side of that graph. With increasing environmental stress, the shorts can't deal with the stress, but the longs can. But let's look at an enriching environment. An orchid really does need tender, loving care to bloom. And when you give that to them, when you grow them in a greenhouse and winterize them and provide them manure and water, they bloom. They look beautiful. Dandelions, on the other hand, don't really care. You can put them in a greenhouse and grow them all you want. They're still going to be pretty much dandelions, and they're probably not going to proliferate all that much more. So that's the story here. We call this the resilience genotype, the longs, because we always associate with lower rates of depression and anxiety. But the truth is that, in a way, being a short makes you more sensitive to your environment. It could actually be a benefit depending on your environmental context. And there is a school of thought that I tend to lean towards, that if you look at the genetic makeup of our populations, the short genotype is fairly prevalent. It's fairly prevalent in both Caucasians and definitely in Asians. And why is that? If it was all bad, if it conferred only risk, then evolutionary genetics dictates that you'd have less of this prevalent in your society. But if you look in the age of man, we have good times and we have bad times. And during the bad times, during war, the dark ages, the longs you could think of as the ones carrying the torch. They are the resilient ones that can deal with war and famine. But in the good times, during Renaissance periods or times of enrichment and enlightenment, it would be the shorts who really bloom, and they're the ones who would carry us forward. Now, again, that's not based on any clinical data. It's really conjecture, but it does make an interesting story. And when you have patients in your clinic and they're looking at their genetic results, it can be somewhat edifying, validating for them to consider themselves, I'm not weak. My stress resilience or the way I deal with stress or my PTSD symptoms are really just a presentation of an unnatural situation. In fact, those with PTSD, the best advice I've ever heard to patients is this: there's nothing wrong with you. Your reaction is to an unnatural situation. It's the situation that was wrong, not your reaction. And if we then understand what the mechanism is at play here, we might provide a better means to mitigate that risk and help overcome trauma. When I talked about replicated studies, this is, again, a perfect example of the difference between an enriching environment and a stressful environment. And this was a randomized clinical trial, the ethics of which could be somewhat questionable, but the data, at least, may be useful. And so these were a little over 100 Romanian orphans. And at the time of this study, orphanages in this country were very poor, underfunded, and usually subject, the orphans in these settings were subject to a lot of trauma. But they randomized half of these orphans to be raised in a high-quality foster care group, which was highly controlled. And they followed them for a matter of 54 months. And they looked at what's called indiscriminate behavior, which you might think of as acting out. It's one of the domains of ADHD, the hyperactivity or defiance. And those who have the long, or the normal genotype, it really made no difference to them in which setting they were raised regarding their indiscriminate behavior. But look what happens to those who have the short genotype. The care as usual, which would be the poor quality orphanages, they had increased rates of indiscriminate behavior, but the inverse was true for those raised in the high quality foster care. They actually had fewer rates of indiscriminate behavior compared to those with a normal genotype. So why is this? I did define what happens with those who have the short allele. It's basically those who have the long, the normal, have normal expression of the serotonin transporter. So they're able to control the serotonergic tone active in their prefrontal cortex and their parietal lobes. And so they're able to control their hyperreactivity or their sensitivity to their environment. But those who have decreased expression of this pump, it generally confers enhanced or elevated serotonergic tone, which creates what we call a hypersensitive amygdala, which is the fear center. It's really the tie between the part of the brain that we think of as us, our frontal and parietal lobes, the things that allow us to interact and sense our environment and plan, and the amygdala, which is sort of part of our primordial brain, our reptilian brain, which is connected with the HPA axis. And we can see the results of this in real time in what's called the Trier social stress test. So these are, this was a somewhat smaller study. This was, I think, about 40 or 50 teenage females. And we see this not just females, but in men as well. But they were at high risk of depression due to family history and environmental context. And they put them in a stressful situation. It was they had to talk in front of an audience, and then they measured their salivary cortisol, which is sort of, you can think of as a measure of stress reactivity, the HPA axis. And those who have the short genotype, their cortisol levels will go through the roof. And so this brings us to the tie between the serotonin signaling in the brain, the part of our brain that we think of as us, and our primordial brain, or the HPA axis, the hypothalamus pituitary adrenal cortex axis. This is a highly conserved system, and by that I mean it's expressed. This is pretty much unchanged in everything from mice to elephants. We all have this system. This is a system that's involved with our fight or flight response, our ability to survive and run away from a tiger, for example. And how this works is, imagine that you're a caveman or a cavewoman, you're out hunting, gathering, and you see a lion, you see a tiger, and you say, oh, I'm going to have to run away. Your frontal cortex triggers the amygdala, say, fear. There's something bad going on, which triggers the hypothalamus to release, the corticoid-releasing hormone. Which then triggers the downstream of sequelae that increases cortisol and adrenaline, which allows your muscles to absorb glucose faster and releases glucose from the liver so you can run away from the lion. If you have a well-developed parietal lobe, you might trip the person next to you so they get eaten first, but that's neither here nor there. So when we talk about anxiety and depression, usually what we're talking about is a dysregulation of this pathway, or misuse. I should say that we, humans, misuse the system. And to describe what I mean by that, I'll tell you another story. If you kick a dog or mistreat a dog or beat a dog, and just to avoid any angry letters, I've never actually kicked a dog. I'm just using this for the sake of the story. If you mistreated a dog and you met that dog 5 years from now, that dog might bite you. because you activated its HPA axis, and it's using it the way it should. It's seeing you as a threat and saying, I'm going to bite you to avoid any threat to myself. But unlike a human, that dog will not have spent the past 5 years fixating on how to get even. That's a very human trait. We're very good at fixating on the past or perseverating over the future, but we're not so good at living in the now. We misuse this organ. And it's likely due to this evolutionary upgrade where we have this sort of hyper-connectivity between the amygdala and the part of our brain that sort of fixates on these thoughts. And another point I might add is I doubt anyone listening to this program has ever had to run away from a lion for survival or fight a neighboring tribe or village for food. We don't use the HP axis for what it was intended. The threats we have now today are things like passive-aggressive emails or mean bosses or emails that should have been meetings or presentations. Today, I can say that my mouth is drying up because I'm a little bit nervous. I can imagine if I measured my salivary cortisol right now while giving this presentation, it would be elevated. And that doesn't make sense because I'm under no threat from the listeners. The worst thing that's going to happen is you're going to fall asleep or turn this off. But I'm fixating on trying to gain approval or I'm afraid of making a mistake. And so that is causing this negative feedback. Now imagine if you had a hair trigger for the HP axis, or if you were constantly barraged with such stressful situations. It's not surprising that this can impact many domains of health. It has been tied in with everything from cancer to obesity, and not surprisingly, post-traumatic stress disorder and mood dysregulation, as well as cardiovascular disease. And I don't know if I mentioned this, but inflammation is very finely tied in with the reactivity of the HPA axis and cortisol levels. So I'm going to talk about two genes that actually act, that directly modulate our stress reactivity. And these two genes would be CRHR1, which is corticotropin-releasing hormone receptor. And so this is the sort of the hair trigger for the fight or flight response. It sort of is responsible for turning that initial signal from the hypothalamus into starting the cascade that starts that sort of chemical cocktail, as Marines call it, as part of that sympathetic response. FKBP5 is actually in the flip side of the coin. It's involved in the negative feedback loop because as you have elevated cortisol levels, that is involved in a feedback loop which starts to turn off the fight or flight response, or what we call the parasympathetic response. So in a way, you can think of this as a trigger for the fight or flight response or a thermostat, and that's what FKBP5 does. It wasn't a very cleverly named protein. It doesn't actually stand for much other than FKBP5, but what it does is it binds glucocorticoid receptor and prevents it from doing its job. It prevents it from going into the nucleus and causing epigenetic changes associated with a fight or flight response. However, in high cortisol environments, so when you see that lion and you stimulate the release of cortisol, FKBP5 is replaced by FKBP4, which brings the glucocorticoid receptor into the nucleus and starts the downstream signaling, which helps turn off the fight or flight response. If, however, you have increased expression associated with a substitution, so C to T, it confers increased protein levels of FKBP5. And basically this is like saturation chemistry here, where if you have a lot of this FKBP5 more than you have FKBP4, it's much harder to dislocate the FKBP5 in a high stress environment, and so it prevents the glucocorticoid receptor from translocating into the nucleus. And you may recall from that first slide that the glucocorticoid receptor, which the FKBP5 protein is binding, is involved in that negative feedback loop. It's not impacting the mineralocorticoid receptor, which is actually causing the upregulation, or that sort of feedback mechanism, the positive feedback, really upregulating that sympathetic response. So what's going on here is you can easily imagine this would cause a very vicious cycle where your brain's saying, okay, this threat is gone, I want to turn this off, but you're lacking this negative feedback loop that help turn off that fight or flight response. And again, we can see this in real time in what's called the Trier Social Stress Test. And what this is very simply put, it's a study of social stress, and it usually involves healthy volunteers, usually college age, because they're willing to do anything for a free lunch. And they put them in front of an audience, and they tell them to do mental arithmetic or do a speech, and they measure their salivary cortisol. Not surprisingly, those who have the increased expression of FKBP5, those with a TT genotype, their cortisol levels go through the roof. Now, imagine if you walk through life with this hypersensitivity to stress. You were always feeling like you had to fly or run away from situations, and what that would do to you. And we actually know what this does to us. So if you are reacting to a lion and you have that bump in cortisol and adrenaline, but then it goes away, that can actually be healthy. That actually enhances immunity and other good health events. But if you constantly are barraged with stressful situations, or in the case of FKB5, if you have a hair trigger, that causes depletion, exhaustion. And when we talk about exhaustion here, generally what we're talking about is depression, PTSD, anxiety. And we see this with these patients. This gene does not cause depression, but in the context of childhood trauma or immediate trauma, it can confer anywhere between a 2-fold up to a 10-fold in one case of depression and anxiety. So again, it's just because someone has the genetic variant does not mean that they're depressed. But then if you look at their situation and their environment, that does increase their risk significantly. Same with corticotropin-releasing hormone receptor, which is like the hair trigger. Those who are raised in a very poor environment, so moderate to severe childhood trauma, so that's sexual or physical abuse, and you measure their salivary cortisol, and in fact, this case, plasma cortisol, it goes through the roof. So it does tie in specifically the impact of the HPA axis with the risk of depression and anxiety. But what can we do about it? If you have this hair trigger, what are some things we can do? Well, I talked about how we're not very good at living in the now. Well, there's very strong clinical evidence, things like meditation and neurofeedback and mindfulness training can not only attenuate cortisol, but it can actually reduce heart rate, reduce the risk of obesity, cardiovascular disease, and markers of inflammation. So I realize that doesn't necessarily have much to do with nutrition, but I can easily imagine that many of you listening to this are in a great position to push or support these modifiable lifestyle factors for your patient population. For nutrition, we can talk about cortisol adaptogens, things like phosphatidylserine, N-acetylcysteine, and ashwagandha. These are cortisol adaptogens, and what we mean by that is these are agents that have been shown to attenuate cortisol in response to stress. In many cases, in the same situation in which these variants confer increased cortisol levels, that Trier Social Stress Test. And so things like omega-3 fatty acids have been shown to both decrease cortisol as well as improve outcomes for depression and anxiety. Phosphatidylserine, which is derived from soy lecithin. Many of you may be familiar with the product Viarin, which basically includes both omega-3 fatty acids and phosphatidylserine. Both of these have been shown to attenuate cortisol. Ashwagandha from Ayurvedic medicine, very strong evidence that it can be a great anxiolytic, and it acts in part by reducing cortisol. And then we'll talk about magnesium, but I'm going to come back to magnesium because the mechanism here is somewhat different than the others that you'll see. And I'll bring in inflammation in a bit. So talking about omega-3 fatty acids, we're used to talking about EPA and DHA. The only difference here would be 2 carbons. DHA has 2 fewer carbons. But they have different impacts on anxiety and depression. And we do know through epidemiological data that those with multiple mood disorders, bipolar, schizophrenia, ADHD, even autism spectrum disorder, we note that they have lower levels of EPA and DHA, both in the serum and in some cases the brain. But that's epidemiological data. Let's look at clinical impact of dosing or providing high-dose omega-3 fatty acids on patients. I did mention already that a high dose fish oil or omega-3 fatty acids can decrease cortisol. In this case, by decreasing cortisol, these patients who are obese actually saw a reduction in their fat mass. So that supports the cardiovascular benefit as well. But what this graph you're seeing is what's called a forest plot. This is a representation of a meta-analysis or a study of studies. And so what they were looking for is clinical trials or clinical studies where they were looking at the impact of high-dose omega-3 fatty acids on depression. So these are patients with clinical depression, MDD, or high scores on the BDI, the Beck's Depression Inventory. And they saw that high dose, and by high dose it was a dose of about 2 grams a day with a high EPA to DHA ratio, was significantly impactful in treating depression. And so if you look at the impact here, the effect size, it's not as effective as maybe some prescription medications, but neither does it have the same risks associated with it. And it's certainly something that you could add on to someone's treatment. Or certainly something you could add on if you thought someone had low dietary intake of omega-3s and they were presenting with a stress reactivity. Again, you're focusing on not just the genetics, but the clinical picture to make a rational decision on their nutrition. Not just for depression, but we see this for psychosis as well. So this was not patients with schizophrenia in itself. These were young adults who were at high risk of developing schizophrenia. So they had psychomimetic symptoms, for example, or they were on the scale, which you might call schizotype bipolar disorder. And so for 12 weeks, these young adults were given about 1 gram of polyunsaturated fatty acids, including EPA and DHA. And then after 12 weeks, they saw that these patients had fewer psychomimetic symptoms, but then they also followed them for 7 years. And so that's what they mean by long-term follow-up. And they measured their symptoms of schizophrenia, which is the PAN score. It measures both positive and negative symptoms of psychoses. And they saw that those who were dosed with omega-3 fatty acids saw an improvement in their symptoms in both psychoses as well as depression, and then their general function. So again, the impact here not as great as maybe some FDA-approved indications, but certainly worthwhile trialing if you have a patient presenting with these symptoms. Similar for ADHD. Now the impact, this is another meta-analysis. That's what whenever you see these forest plots with basically these are the author names, they're looking at this is a study of studies, and they look at the average response rate for omega-3 fatty acids to treat ADHD. And they did see that those who were treated with omega-3 fatty acids did see an improvement in ADHD symptoms. Phosphatidylserine, similar, basically a good segue from talking about ADHD with omega-3s is phosphatidylserine, which is often used as a nutraceutical for ADHD. But I would purport that it's even better tailored for those who have hyperactivity or problems dealing with stress in their environment. And so we have multiple studies showing that about 400 milligrams a day of phosphatidylserine has been shown to decrease cortisol in response to stress. Ashwagandha from Ayurvedic medicine, across multiple measures of both depression and anxiety and general quality of life, has been shown to improve outcomes and has been shown to directly modulate cortisol significantly. This is almost a threefold decrease in cortisol levels compared to those on the placebo. Now, I just sort of did a quick run-through, and I mentioned a few items. I mentioned omega-3 fatty acids, phosphatidylserine, ashwagandha, and magnesium. And it's not surprising that agents that have been shown to attenuate cortisol reactivity also have anti-inflammatory properties. I did sort of foreshadow this earlier in talking about how the HPA axis is finally in tune with inflammatory pathways. And so I mentioned a few agents that you can use for stress: ashwagandha, magnesium, omega-3 fatty acids, phosphatidylserine, and that's just to name a few. And it's interesting, I have noted that many of these agents that act as cortisol adaptogens, which means that agents that have been shown to directly attenuate cortisol response to stress, also have anti-inflammatory properties. There is a very close link between our regulation of the stress response and inflammatory mediators and inflammation. But the mechanism by which some of these agents confer that impact on cortisol may follow an inflammatory etiology, because not only does activation of HPA axis cause inflammation, but inflammation has a negative feedback loop that feeds back into activating the HPA axis. But there's a missing link here. So we tie inflammation with neurological issues, psychoses, Alzheimer's disease, depression, anxiety. We know that there is an inflammatory etiology here, but it's hard to pin that down. One of the low-hanging fruit as far as a target for that tie-in was the HPA axis, but it seems that it's more central to that. It's actually less to do with the HPA axis, more to do with the part of the brain we think of as our cells, the actual neuronal excitation itself, particularly hyperexcitability of the glutamate pathways. And I think this is a great representation or illustration of this. So what you're seeing here is this Venn diagram is another meta-analysis. And this was a meta-analysis not of clinical trials, but of what's called genome-wide association studies. And genome-wide association studies are really just gene studies where they look at patients with a disorder and compare the genes or the genetic variants of people with a disorder with those in the normal population without the disorder, and then see what the difference is. And so you'll come up with what are called candidate genes for diseases. And they're interested in what are the pathways associated with autism spectrum disorder or bipolar. But these authors, the Lotan's group, what they did is they did a meta-analysis and they looked at what is the overlap of these different genome-wide association studies of ADHD, schizophrenia, depression, bipolar, and autism spectrum disorder. And 15 genetic markers popped up. And of those 15, 5, ANK3, CACNA, this gene here, CACNA1C, and these two genes, so 5 of the 15 are what's called ion channels, which are expressed in neurons. The two I'm going to discuss with you is ANK3 and CACNA1C. So ANK3 is actually an anchoring protein that controls how the sodium channels in the nodes of Ranvier are placed. And CACNA1C is the voltage-gated calcium channel that's expressed at the axon terminals. And so how these work is, you see a mouse and you shriek and you want to activate the part of your brain that, activates fear, for example. I just kind of threw that out there. But, the presynaptic neuron releases glutamate. Glutamate hits this ligand-gated sodium channel, which allows sodium into the cell. That causes a depolarization, which then propagates a signal through the nodes of Ranvier in these voltage-gated sodium channels controlled by ANK3, and finally hits this voltage-gated calcium channel, which allows calcium into the cell, which triggers something in the cell, usually the release of a neurotransmitter. The variants that were associated with these multiple mood disorders confer dysregulation of these ion channels. The T allele of ANK3 confers altered clumping of the sodium channels in the nodes of Ranvier. The A allele of the calcium channel confers excess calcium influx. So in other words, these neurons are just hyperexcitable and they're just firing all the time, or they're dysregulated. And so it's not surprising that it's associated with multiple paroxysmal disorders. What's also not too surprising is that agents that have been shown to be cortisol adaptogens, things like magnesium and acetylcysteine omega-3 fatty acids, one thing that they also have in common, besides having anti-inflammatory properties, is they block or attenuate this excitatory signaling. They attenuate sodium influx and they attenuate calcium influx. Magnesium in particular is a natural calcium channel blocker. And so one other association that we've seen with calcium, the CACNA1C variant, the voltage-gated calcium channel, is this excess calcium influx is also associated with sleep latency, inability to fall asleep, which shouldn't be too surprising, because if you have a hard time letting go from the day because your neurons just keep on firing, it's not surprising we'd have trouble falling asleep. And I'm sure we've all had nights like that. Well, magnesium, as a natural calcium channel blocker, has actually been shown to promote healthy sleep, to improve sleep duration and quality. It acts similar to prescription drugs that have recently found utility in depression and PTSD. Magnesium, as I said, is a natural cortisol adaptogen. Patients who were actually given a high dose of magnesium, so this is a little over 200 milligrams of elemental magnesium, which would be like 4 tablets of a 500 milligram magnesium chloride tablet, they actually saw improvements in depression scales over 12 weeks. So again, it's nowhere near the impact of prescription medications, but certainly is something worth adding on, particularly in our Western diet, which is usually lacking in magnesium. And so this is one way you might interpret this. So the voltage-gated calcium channel is associated with altered neuronal signaling, excess calcium influx associated with the sleep dysregulation, mood lability, depression, anxiety. And we know the mechanism here. We understand the mechanism at play. And so by targeting that mechanism with things like omega-3 fatty acids, particularly if they're presenting with stress hyperreactivity, is a rational agent to add on, as well as melatonin and magnesium. The next agent I'd like to discuss is brain-derived neurotrophic factor, BDNF. And so this is a protein that is ubiquitous throughout the body, but is particularly important in the brain in promoting what's called synaptogenesis, or neuroplasticity. You can think of it as miracle growth for the brain. This picture you're seeing here is actually a rat neuron bathed in a dilute solution of BDNF, and you can see after 48 hours, this neuron really wants to get touchy-feely with its neighbor. It just wants to grow and connect and form new connections. BDNF is actually expressed as two proteins. When it is first made, when you go from the DNA to the RNA to the protein, it's expressed as what's called a pro-BDNF protein. And this protein actually is involved in breaking down those synaptic connections. And this is important. So, when we're born, we have, lots of neurons in the brain, but they're not really doing much. And as they start to grow, they make these connections, but not all those connections are really important. And around the age of 6, you have a lot of these, connected neurons, but they're not really, doing anything. Anyone who has a 6-year-old or has raised children can attest that, these when they're around the age of 6, they're not very rational beings. And this would be why, is they have these connections in the brain that are important. So pro-BDNF is important because it prunes those synaptic connections. But we also want to strengthen the connections that are important for, say, learning and memory and mood. And to do that, this pro-BDNF region is cleaved to create the smaller region, which actually is active in synaptogenesis. The variant that we're looking for is actually called the Val66Met, which is really just a short name for at the 66th residue or 66th amino acid, because proteins are made of multiple amino acids. Instead of a valine, which confers a rigid structure, an active structure, some people will have a methionine, a met. And that met actually makes somewhat of like a floppy, a soft protein, if you will. And if you have this, change in the shape of the protein is less able to cleave off the pro-BDNF domain and make the mature BDNF region, which is responsible for sustaining those connections. Not surprising then that those with a Val66Met gene, or the variant, they often have impaired working memory. Working memory is usually what's called, they give you a series of numbers and you're supposed to repeat them back. And in general, those with a met allele score worse than those with a Val. It also impacts stress response. So that trier social stress test, patients with a mental ill generally have higher cortisol levels, which is not surprising. It is also associated with depression. But the good news is this. This is less of a stress diathesis model following gene and more of a differential susceptibility. And this is what I mean by that. If you compare the VAL-MET or MET-MET carriers to the VAL-VALs and test their working memory, so this is that test I was talking about where they give you a series of numbers, generally those with a MET allele score poorer or have lower scores than those with a VAL-VAL. But if you put both of those subjects on an exercise regimen, not only will those with a MET allele compensate for that reduced activity, they'll actually exceed the scores of those with a normal genotype. And why this is I think I mentioned earlier that this is a ubiquitous protein. It's not only active in the brain, but also active in the body. And skeletal muscle is involved in upregulating BDNF. So by increasing skeletal muscle movement, and we're not talking about Olympic weightlifting here or marathon running. It's at least 3 days a week, 30 minutes with 80% of your maximal heart rate. So most patients can achieve this, but the truth is the more the better. You can compensate and actually exceed and reverse the impact of the variant. Now, in addition to exercise and lifestyle modification, the diet plays a role as well. And so what you're looking at is the results of a preclinical study in rats where they fed these rats high fructose corn syrup or a low fructose concentration control, basically a diet that had less sugar. And what they saw is that the rats who were given the high fructose corn syrup, their levels of BDNF were significantly lower than those who were given the more nutrient-dense diet. And that nutrient density, that's important. So this is preclinical, but we can relate this to humans as well. We know that those who have a high fat, high sugar diet tend to do poorer in school and in life than those who have a nutrient-dense diet. And one of the reasons this might not be due to sugar and fat specifically, but maybe due to the other things that you'll find in a nutrient-rich diet, such as dietary fiber, a diet that supports our microbiota. In particular, butyric acid is a short-chain fatty acid that is made in huge quantities by our good bacteria in response to dietary fiber. These short-chain fatty acids do lots of things in the body, but they are very potent epigenetic modifiers. They inhibit HDAC proteins, which are histone deacetylase, which are really the things that control gene expression. And there is a huge body of evidence supporting the fact that these SFCA, these short-chain fatty acids, such as butyric acid, directly modulate or upregulate BDNF. So what I've done is I've tied the brain, our central regulating part of the brain, our frontal cortex and parietal lobes, and these catecholamines that help us, be who we are, into the stress response, which is integrally tied to inflammation, as well as our brain-gut axis. By supporting our gut microbiota with things like dietary fiber, you can actually directly modulate these BDNF proteins, which is like miracle grow for the brain. And so how you would interpret this is those who have the MET allele, the number one recommendation, very strong clinical evidence, is physical activity. And then look at their diet as well. So dietary omega-3 fatty acids, actually I hadn't mentioned this, has been shown to enhance BDNF levels as well. And then we'll talk about the preclinical evidence for saturated fat. Now, I have not mentioned lithium, and this might be somewhat of a touchy situation because when you think of nutrition, you don't generally think of lithium. Whenever you think of lithium, you think of the high-dose lithium given to bipolar patients in grams at a time. But we have seen in both clinical and preclinical studies that lithium enhances BDNF. And there's data that shows that microdose lithium, so 300 micrograms lithium, which you'd find in something like lithium orotate, for example, or in your diet. You can find this in tap water. and actually is ubiquitous. It's in the soil, it's in our water, and similar to magnesium, has been depleted from our diet, from the foods that we eat over the years. And they actually have data that I'm not showing here that implies, I should say, it's not very strong, clinical data, it's epidemiological, implies that in areas that have higher elemental lithium in the drinking water, they have lower rates of suicide. We do have, what you're seeing now, a randomized control trial of patients with Alzheimer's disease who were treated with microdose lithium, the 300 micrograms lithium, over a course of 2 years, and then measured, I don't have it listed here, the MMSE, the Mindy Mental State Exam, which is a measure of their cognition. And those who were on placebo saw, or they displayed, the normal decline in cognition that you expect with patients with the pathophysiological signs of Alzheimer's disease, but not those who were treated with a microdose lithium. And by tying that in with a mechanism that it enhances BDNF, supplies one possible mechanism. Another is that lithium may actually act as an anti-inflammatory agent in its own right by inhibiting or downregulating excitatory signaling. Now, it's a little ways to the well on this one when I talk about how lithium might be an anti-inflammatory agent. It's somewhat indirect, and it goes into how I was referring to the calcium and sodium channel as the missing link. The variants associated with mood dysregulation confer excess excitatory excitation, so signaling. That causes lipid mobilization, which increases arachidonic acid, which is directly linked with inflammation. It also increases extracellular glutamate because one of the neurotransmitters that's released is glutamate. And glutamate outside of the cells increases NO synthase, nitrous oxide, which leads to free radicals. By inhibiting those NMDA receptors, you are shutting off this pathway in essence. So as summary, I talked about just a few of the genes that impact stress response and other domains of health that are intricately tied in with our response to stress and mood regulation. But the focus, of course, is always on the mechanism by which these variants confer that risk. That provides an opportunity to target that pathway for things like inflammation. If you have a patient who is definitely displaying signs of stress reactivity and they have a variant that confers that risk through hypersecretion of cortisol, we already talked about a few agents that have been directly shown to inhibit cortisol. So if you found this discussion enlightening today, or if you think you'd be interested in using personalized nutrigenomics in your practice, the genes that I discussed today, as well as several others, are included in a genetic assay designed for just that purpose, called Mindful DNA Professional. The focus is to look at a systems biology approach, to take a systems biology approach to wellness, to look at these pathways, and to focus on the mechanisms by which these genes, 32 variants that we look at, cross domains of health, and how by looking at the mechanisms involved, one can improve the wellness of our patients using lifestyle and dietary intervention. For more information, you can visit the URL that you see on your screen, genimind.com/mindful-DNA. So I don't know, Meredith, if we have time for questions, or if there are any questions. I couldn't actually see them on my screen.
Yes, definitely. Thank you for that wonderful presentation. And yes, we do have time for a few questions. So first question, you mentioned the link between inflammation and psychiatric conditions. What are your thoughts on the use of hemp as an anti-inflammatory agent to target depression and anxiety?
Okay. So hemp is actually widely available as an over-the-counter. Now, the active ingredient would be a compound called CBD. I imagine everyone listening knows what CBD is. And actually has been shown to have anti-inflammatory properties, mostly in preclinical models. It is not considered, the CBD itself is not considered to be a dietary supplement in itself. But it's actually been shown, at least at high doses, to be effective It's actually FDA indicated for a specific type of epilepsy for children. But when I say high doses, this is in hundreds of milligrams for small children and is actually associated with adverse effects. At the doses that you'd see in many of the over-the-counter hemp oil type products, which is in tens and tens of milligrams, it's unclear if it's effective for the same indications or treating specific disorders. But the truth is, we do know that it does have anti-inflammatory properties, and there is a lot of preliminary evidence supporting its use in anxiety, case reports and things like that. But to date, there's no large clinical trials showing efficacy. But at the low dosage that you'd see in things like hemp oil, natural hemp oil, not the purified CBD, it's generally considered safe for use. So I think, again, it falls into the same category that when I was discussing omega-3 fatty acids and magnesium for anxiety, depression, PTSD. It won't have the same effect size or same potency as, say, prescription medications, but neither do they have the same risk profile. I don't know if that answered the question, or if I can get feedback from the questioner.
No, that's perfect. Actually, very interesting. Thank you. You mentioned many supplements that might be used, but as a nutritionist, my focus is primarily on the food choices my patients make. Can you provide any insight on diet with any of the genes you talked about today?
Oh, okay, yeah, absolutely. Well, in general, I imagine, I don't know if I said it during the discussion. I probably should have, knowing the audience, but for the most part, wherever we're making recommendations for specific nutrients, if one can attain it in the diet, then of course it would be preferable for many reasons than supplementation. We know that usually the salt formulations for magnesium chloride, for example, or even omega-3 fatty acids in that purified form, the absorption and its utility is not as potent as when we attain these same nutrients in the diet. In fact, I would say for omega-3 fatty acids would be a perfect example. Most of the data supporting omega-3 fatty acids for cardiovascular health and anti-inflammatory benefits, it's actually based on epidemiological data where they showed that those who have a high intake of fatty fish, or those who have a high adherence to the Mediterranean diet, for example, have a lower risk of these outcomes, such as cardiovascular disease or Alzheimer's disease. So yes, absolutely. Many of the supplements I discussed today, if you are giving dietary counseling, then you can, I'm sure the listeners to this program know better than I the specific foods to look for magnesium, for example, the dark leafy greens and fatty fish and things like that. I don't know if that answered the specific question you were asking about genes. So we don't have any direct link to the genes to diet, but as I said, when we make these mechanistic connections between things like magnesium as a natural calcium channel blocker, and you're looking to address maybe sleep latency or anxiety, then besides adding a supplement, you could look at diet. You could look at increasing their intake of dark leafy greens, or look at agents, supplements that, instead of using a purified salt formulation, look at basically compounding or sort of providing a formulation that is more natural or more similar to what we attain in our diet.
Excellent, thank you. And you touched on magnesium a bit. What dose of magnesium would you recommend for anxiety?
Okay, well, in the studies that showed, at least the preliminary studies that showed efficacy for reducing self-reported anxiety and self-reported depression, it was actually a fairly high dose. And by high dose, they for the most part, they used magnesium chloride at a dose of 500 milligrams or above. And why I call that high dose is that's when you get up to around 500 milligrams, that's where you do run the risk of diarrhea and loose stool. And why that is magnesium is in these, actually it goes to the previous point when we were talking about dietary intake over supplementation. When you take this high concentration of magnesium salts and it gets into the GI tract, it takes a little while for it to absorb. And when magnesium just sits in the GI tract, it absorbs water. It actually pulls water out of the body and causes local irritation, and that's how you get stool. You'd have to be very careful then for many of your clients or patients you're seeing where you're looking to address anxiety. I would also assess whether or not they're taking prescription medication for anxiety. Usually these would be serotonergic agents, SSRIs, SNRIs. And one of the well-known side effects of these agents is they actually increase the risk of diarrhea, because a lot of the serotonergic neurons and serotonin receptors are actually more prevalent in the gut than they are in the brain. And so in those cases, I think the age-old quote or adage of starting low and going slow is always useful. The recommended daily intake of magnesium is around 400 to 600, depending on whether someone's pregnant or how large they are. I would start maybe 250, 300 milligrams, and different salt formulations actually have different risks of diarrhea. So magnesium gluconate, magnesium glycinate, to some extent magnesium threonate are generally thought to have a lower risk of diarrhea than other formulations. But again, I think if you're as long as you're keeping it in the lower end of the dosage, maybe splitting doses throughout the day. If you're going to give a high dose of 500 milligrams, maybe split that between morning and evening. I don't know if that answered the question or not. The truth is there's many different formulations of magnesium, and there are good resources out there that you can look to find more information on those risk of diarrhea or efficacy.
Okay. Thank you. Alrighty. And last question, there's a clear link between inflammation and dementia. Based on your discussion on how inflammation is linked to stress, do you think there is a link? And if so, what is it?
Okay. So actually that the way the question was worded, I actually don't need to ask for clarification whether I think there's a link between inflammation and dementia, inflammation and anxiety, or anxiety and dementia, because the answer is yes for all three. So I focused actually during this talk on stress and anxiety. What I didn't talk about, because I wanted to remain in scope, is many of the interventions that I discussed are also effective or have been shown to have efficacy in reducing the risk of dementia. And if you look for the link between inflammation, stress, and dementia, we can look at actually other genes that would be on Mindful DNA panel that are on there because they are associated with the risk of dementia, a risk of Alzheimer's disease. Now, again, as I said at the beginning, that doesn't mean they cause disease. They're just merely associated. And we understand the mechanisms of some of these genes. They're actually triggering neuroinflammation in the brain. And some of the interventions I discussed today, such as lithium, has been shown to reverse the impact of these mutations when they're actually when we use them as disease models in animal studies. I think just to clarify, actually, the gene I'm thinking of is TREM2. It's a molecule that actually triggers or controls the neuroinflammation in the brain caused by the microglia cells, which are cells in the brain that you can think of as immune cells. And what we do in animal studies is we'll actually knock out that gene. We'll actually make a mouse that does not express that gene, and sure enough, that mouse will have dementia. They'll have the pathophysical signs of Alzheimer's, including the plaques that form in the brain. But when these same mice were treated with lithium, it was shown to actually ameliorate the effects of this loss of function of this gene. And the variant that we look for on the panel is actually a similar thing. It causes decreased expression of
this gene. And so when I discussed BDNF and how BDNF is enhanced by microdose lithium, there would be sort of a gene-gene interaction there that would support that intervention. If we look at omega-3 fatty acids and magnesium, for that matter, I mean, to tie in inflammation, the same, the same connection I made between stress and inflammation for anxiety also applies for dementia. If you have activation of the HPA axis, either through horrible stress throughout life or prolonged stress, or if you have a genetic predisposition to dysregulation, that is actually going to be associated with increased neuroinflammatory response, which is an independent risk factor for dementia, for those the pathophysiological signs of dementia. And so the truth is, there's no single answer to say this is what we can look at that shows that there's a link, but we do know that many of the same pathways that are associated with anxiety, depression also are involved in cognitive function. I don't know if I just ran on there forever or if I answered the question appropriately.
No, it's really fascinating information. It's really wonderful. Thank you. And unfortunately, our time is up today. I think this has been an enlightening evening. Thank you, Dr. VanDorn, and best of health to you all.